Hihinanta natin Pabukal sa iyong isipan, tagumpay na tatahakin Isang pangarap na lubos, landas na iyong pagtutunguhan Pangat na inaasam, di mapipigil ni numan Maaabot lahat, lahat ng iyong inaasam Kasipagan ng sabot, salat ka man sa kayamanan Buksan ang pahina ng mga aklat ng karunungan Edukasyon ating kailangan, kamamangan ating iwanan Peace!
Ating lilipanin ang himbawawin
Ating tanawin
Bagong pag-asa ay muling tatanawin
Kinabukasan pagtibayin Pilitin mong gawin
Puspos ang pagsisika
Pag-asa ay yakapin Edukasyon ang kayamanan ng kahit liman Isang pangarap ang tutupad Magsisikapan ng lahat
Puspos ang pagsisika
Kaya mo yun, kaibigan
Mabubuo isang
Knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na! Do you want to gain knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na! Do you want to gain knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na! Do you want to gain knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na! Do you want to gain knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na! You want to gain knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na! Do you want to gain knowledge and skills from upskilling events? Matutulungan ka ng DigiField Tech Nation. Nag-o-offer ng free webinars like Research, Adobe Photoshop, Microsoft Trainings, Google Tools Training, and many more. Made by teacher for teachers. Katuwang ng mga guro. Join na!
Okay, so good morning, everyone. Thank you for all the viewers and to all the teachers watching right now for this session for today's webinar. Okay, so inviting everyone to like, share, comment, and subscribe this channel for you to be notified for upcoming sessions. Okay, under this channel Digital Tech Nation. Okay, so our session for today's event is a very interesting one, especially if you are in the field of science. Okay, okay, to give a guide, to give inputs to our educators and our learners on how to make this what we call as the science investigatory project. Okay, so without any further delay, okay, so let me introduce our guest speaker for this morning's session. Okay, so our guest speaker for this morning session is science teacher for 17 years. He received the diploma course for research and development of products to meet public health needs at Tropical Medicine Nagasaki University in Japan in 2012. Okay, he has been a winning coach in science investigatory project. His team also won a gold medal for the research project on Sagip, a mobile and portable IoT-based weather station during the 7th World Invention Creativity Olympic or the WICO 2018 in Seoul, Korea. Because of his invaluable contribution to teaching innovations, the Department of Education awarded him the ICT Achievers Award during the DepEd Cyber Expo at PICC Pasay City in March 2019. Robotics and Arduino's programming motivated him to develop a weather wiser. an EU-based monitoring station which won him a gold award again in the 4th International Invention Innovation Competition in Canada, or ICANN 2019. He then presented also to improve the version of the innovation called Wiser, which garnered a silver award during the prestigious Advanced Innovation Global Competition at Nanyang Technological University in Singapore in November 2019. Invited to the USA-based International Virtual Science Symposium, or IVSS 2020, and was one of the panel judges to score students' research entries around the world. Some of his many accomplishments include his two publications in an international scientific journal, Antioxidants and Cytotoxic Activities of Select Endemic Plants Against Lung and Colon Cell Lines, an assessment of the genotoxic and cytotoxic responses of colon cancers, cell lines on white oyster mushroom metabolites. A proud UST scholar and alumnus of Ateneo de Manila University, obtained his Master of Science in Science Education on August 10, 2020. He is currently teaching high school biology and earth science in San Francisco, California, USA. He has been a volunteer judge in First Tech Challenge and Robotics Leagues in Northern California. Our speaker also believes that education must be useful and relevant. In addition, he thinks that innovation in education encourages teachers and students to be creative and forces them to approach problems from higher level of thinking. Once again, our speaker for today's session is no other than Mr. Saldi Diaz Alima. Sir Saldi, good morning, sir. Okay, how are you? Okay, okay, naka-mute po kayo, sir.
Sir, ako mag-i-on internet, sir.
Okay, sir. Go ahead, sir. Naka-mute po, sir. Okay, while nag-fix po si sir sa kanyang technicalities, again, everyone, invite your colleagues, your co-teachers, and other science enthusiasts, okay, to join this session for our introduction to science investigatory project. Okay? So, may kaunting problema lang si Sir Saldi sa kanyang technicalities. Okay? So, okay. Okay, once again, Dr. Saldi Diaz Alima. Once again, good morning, sir. So again, dear viewers, this session is intended for educators and students, okay, wanting to learn about introduction to science investigatory project, okay? Kasi alam naman natin with regards to science, maganda rin maturuan natin yung mga learners natin in terms of basic research, okay, application of some available resources that we can use in our environment. Okay? So nawala si sir. So, kaunting ano lang, kaunting glimpse lang sa background ng speaker natin. So, ang speaker natin ay graduate from Ateneo de Manila University po. Dati pong public school teacher din dito sa Philippines. But ngayon, he's currently teaching sa California po. Okay? Teaching biology and earth science in California. Okay, high school po ang dinadala ni sir. and also one of invited judges po sa iba't ibang mga activities ng science doon sa US. Okay, once again, good morning, sir. Okay, pasensya po, sir, sa technicalities. Sige po, go ahead po. Pakilakasan lang po ng audio po. Okay? So by the way, viewers, with regards to time zone, magkaiba pong time zone natin ngayon from our speaker kasi si Sir nasa US ngayon. So we are daytime ngayon dito sa Philippines. So sa kanila iba po yung oras nila ngayon. Okay, so still we're so thankful na binigay ni Sir ng kanyang time despite po sa okay, not yet, sir. Wala pa pong audio po. Okay. So again, sa mga viewers natin ngayon na nakatutok, kindly invite your science teachers, your students, especially handling po ng research po. Okay, napakaganda ng topic natin ngayon because it's about science investigatory project po. Okay? So alam naman natin na mga nagsilabasang mga gamot ngayon, nagsilabasang mga products, okay, process, tools, nagsimula po yan sa science investigatory project po. Okay? So by virtue of trial and error method, okay? So testing the hypothesis, okay, is it applicable to human po? Nasa-solve ba yung problems, needs? Okay, so yan po ang isa sa mga dahilan po bakit nandito ang Science Investigatory Project. Okay? Kasi way back po sa, balikan natin yung tree of science. So there are three main routes po. So we have curiosity, problems, and needs. So by virtue of those three routes po of science, so yan po ang isa sa pinanggalingan bakit nandito po si Science Investigatory Project. At in-apply po to from elementary to high school. Okay, sir? Okay na po ba, sir? Nakamute po kayo, sir. Ayan. Okay. Sige po. Sana po okay na po yung audio natin. Okay. Okay, and also shout out to my classmates in masteral units since may kasama po tayong mga ano din nagma-master ng science kay Maed in teaching physics po. Okay? So shout out po sa inyo sa session natin. Okay? Malaking bagay po ito binigay ni Sir na for free for us. Okay? So siguro, sir, okay na yung audio natin. Okay, di pa po nakapasok yung audio, sir. Sige, sir. Audio check po, sir. Wala pong audio pa. Okay. So sana makapasok na si Sir Saldi ang kanyang audio. Okay. Kasi again, ang ganda ng topic natin ngayon. Okay. So again, what's out to my classmates in master class in Cebu Technological University dito po sa Cebu City, Philippines. Okay? So,
Am I audible now?
oh yes, ay, okay. Yan po, nakapasok na po sa wakas. Okay, so
Ah,
pasensya po sa technicalities po natin.
yeah, so I
Okay, sir.
use
Sige,
my
sir. Go ahead po.
iPhone microphone. Okay, so let me present slides.
Okay, so kumusta po tayo dyan, sir? Anong oras natin dyan sa California ngayon?
So right now it's around 7 p.m.
Okay. So,
Can you see my screen?
nakapaghapunan na po ba tayo, sir?
Oh yeah, sir. Can you see my
Okay.
screen?
Okay, so sige sir, share your screen po para mailagay natin sa live broadcast natin.
Okay, so can I just send my slides to you, sir? Kasi I can't.
Sige sir, so I will put my ano sir, through email sir or sa messenger na lang.
Here, I'll send it here.
Or else I will give my email po sa private section natin.
So I'll send to you in
So
your
sa
line.
private chat natin, lagay ko po ang email.
Okay, we go.
Okay. Okay, let me check. Okay.
So I try to present screen sharing.
Okay, so nakita ko na po and downloading na po. Okay. So at least po hindi po masyadong kakain ng bandwidth kasi kung gamitin natin sa online po baka hihina yung ano natin. Okay.
Okay.
So almost done na po. Okay. So sir, ano lang sir, pahapyaw lang sa mga nanonood natin ngayon. So ano kayang pwede natin na ma-expect sa topic natin, an introduction to science investigatory project. Gaano ba kahalaga yung ano natin? Science investigatory project. Sige, go ahead po. Okay, nawala din audio ulit sir. So walang audio po.
How about that? Okay, I'm back.
Yan. Yan po. Okay.
Okay, so you can just, ano na lang, sir, present my slides.
Yes po. Okay, so allow me to share my screen. Okay, for a while lang, dear viewers, while fixing for some technicalities. Okay. Okay, for a while po.
Meron ba tayong mga students, sir, ngayon watching us?
May mga stories tayong nakatutok ngayon, sir. But ang ano lang, sir, yung sa class ko din ay yung iba, sir, ay sa team replay kasi sa school namin ngayon ay car day. So yung iba ay galing pa dito at pauwi pa po ngayon. But some of the teachers watching right now, especially may kasama ko po sa masteral class ko. Okay, so nanonood sila ngayon. Okay.
I'm sorry for the technicality.
Ah, okay lang po. Okay, so again, share screen. Okay, so ayan na po sir, naka-flash na po sa screen natin.
Okay, here we go. So, good morning there in the Philippines. It's around 7 p.m. here in San Francisco. So, welcome to our session on science investigation. So, I hope that students, teachers will be able to learn from this webinar. Okay, next slide, sir. Do you have, okay. So, the topics that we'll be covering today
Ayan po.
are following. So, number one, so what's the reason why we conduct science investigatory projects, specifically students who are studying in biology, physics, chemistry, earth science. And then we're gonna cover also some overview of scientific method, and then we'll discuss a little bit about CARS model, especially in writing introduction, and then we'll talk about finer in constructing research title, then experimental design, the use of PICO method, and then we'll also talk about some animal models used in lab and in some IC forms and writing tools. Next slide, sir. So like what I said earlier, science investigatory is very well encouraged in the Department of Education. So we consider science investigatory project as an avenue for teachers and students to learn more about science through investigation, through laboratory experiments, so that they, especially the students, acquire knowledge in science, apply concepts in science, theories, principles, and laws of nature. And of course, the HOTS, the higher order thinking skills, will also be developed by our learners. Okay, so this is the overview of the scientific method. So we always start with making observations, and we ask ourselves, what do we see in nature? This can be from your own experiences. It can be also from your own thoughts or based on your reading. And so from making observations, we ask interesting questions. So why does that pattern occur? Or what pattern we see in nature? And then we formulate hypotheses. Um, we ask ourselves, what are the general causes of the phenomenon we are wondering about? And then we develop testable predictions. We perform experiments. We gather data, and then we'll go back to making observations. But this process of scientific method is not actually a one cycle. So as you can see here, it's a cyclic pattern. But in real science, it doesn't matter which steps or process you start. There are a lot of innovations, inventions that don't start with observations. Some are the results of like serendipity or just accident. So we'll be talking about that later on as we go with our presentation today. So please, next slide, sir. Okay, so we have 6 here, main steps in science investigation. So the first one, the next slide, please. Next slide, sir. Okay, so the first step is choosing the topic that you're very interested. So for example, here, if you're interested in culture or making culture of bacteria, if you're interested in analyzing the chemicals from the plants, we call that bio chemicals or phytochemicals in most of the plants. If you're interested in finding solutions or like treatments for different diseases, you can also use this as your avenue for investigation. And then if you're also interested in smart machines or artificial intelligence or robotics, that's also a good topic to pursue. Next slide. So when we conduct research, it's very important that we know where to get the right information. So I have posted here some references, some scientific journals that are free. So number one here, which is very available and free. We call this eric.ed.gov. So if you click that link here, this will land you to uh, a website and allow you to download some of the PDF files that you can use. These are free. And then the common one is the Scholar from Google. And then we have so I hope I can click this file here, connectedpapers.com. So for the connectedpaper.com, this one is, um, so please click that file, sir, if you can click that link. Okay, so we'll just proceed to this slide. Okay, so the next step would be developing your hypothesis. So hypothesis is a statement that explains expected outcome of your experiment, and usually it is based on your research and your understanding of the topic. So earlier we have talked about choosing your own topic of your interest. So now you do research and then develop your hypothesis. So there are kinds of hypothesis here. Number one, null hypothesis. So it is a hypothesis of no difference. There's no difference between two statistics or between statistics and parameter. And you also have alternate hypothesis, which is a hypothesis of difference. So there is difference between two statistics or between statistics and parameter. And then the very common kind of hypothesis that usually students and teachers use in the classroom is the conceptual hypothesis. So it is a general statement, not in terms of statistics, of relationship between dependent and independent variables. Next slides. Okay, so now when you're ready with your hypothesis, we have to perform the experiments. We design and conduct the experiments. So in this step, you'll design and conduct experiments to test your hypothesis. And then, of course, when you conduct your experiments, there are variables that you need to be familiar with and also identify the controls and collecting data. Next slides. So it's very important that when you collect information or data as you perform the experiment, you need to have a laboratory notebook. So it is a permanent record of what you did and what you observed in the lab. And this includes the mistakes and difficulties performing the experiment. Sometimes some things um, won't won't go the way we expect them when we perform experiments. So this is a good avenue for you to record your data, your information from the experiment, and it is very useful, especially protecting your intellectual property. For example, if you're applying for patents, and Lab Notebook is actually uh, a good opportunity for you to make ownership or authorship of your discovery. So between gaining or not gaining recognition, laboratory notebook is a piece of evidence for you to support your discovery or innovation. So on the right side, we have here parts of the lab notebook. So you have the introduction, you have here the purpose, why you conduct this experiment. On the upper right side, you have the date, and then you can also paste here at the center the the protocol or the step-by-step procedure in your experiment. And then you can also include here your measurement, the data that you want to generate, and also the purpose and the expected outcome. And then this lab notebook has to be signed, and it should be dated on the day you perform the experiment. Next slide, sir. So when you have already collected your data, you have recorded all your information in the lab notebook. So you can now tabulate your information, your numerical data, and ready for analysis. So after collecting your data, you have to interpret the data, determine if your hypothesis is true or if your hypothesis is correct, or you have to accept or reject your hypothesis. And then this information can be presented in a form of graphs, in a form of tables, and other tools to make sense of your information or your data. And then we have here draw conclusions. So based on your analysis, you'll draw conclusions about your experiment. So if your hypothesis was supported, you can explain why, and if it wasn't, you can discuss possible reasons why. Next slides. Okay, so now let's talk about Carl's model. So Carl's stands for Create a Research Space. So this model, the Carl's model, was developed by John Wells based upon his analysis of journal articles representing a variety of discipline-based writing practices. So this model attempts to explain and describe the organizational pattern of writing, especially the introduction to scholarly research studies. So how can we use the Carl's model? So we have here number one, begin the writing process, and especially that getting started is a very difficult step for you when writing your paper. And then number two, understand why, understand the way in which an introduction sets the stage for the rest of your paper. And number three, assess how the introduction fits the larger scope of your study. So let's tackle more about this. So you have different moves. So let's start with number one. Move one, establish a research territory. So the research territory or the broad topic is the context required to both understand and conduct your research being explored. So it tackles also the goal and explain the concepts, and you ask this question: why is this general research area important? And then you have here at the bottom here sample statements or language in establishing a research territory. So for example, here you want to study on types of bacteria present in soil. So you can have bacteria has in soil has been extensively studied in blank, and so and so. An interest in different strain of bacteria in soil has been growing, or recent studies have focused on the study of bacteria on different soil types. So these are examples of statements that demonstrate establishing a research territory. And basically, this area here answers the question why it is very important. The number two is establishing a niche. So the niche is the reason or motivation for the research. And in here, you highlight the gaps or the problems and also the current knowledge. So we have some methods to use in establishing a niche. So number one here, we have make a counter claim. Something is wrong. What is wrong in your chosen topic? Indicate a gap. So what has been missing in this area of study? And then raise some questions or make an inference that something that is not clear in your area of topic, and then continue a tradition or adding something. So that's our method of establishing a niche. Next slide, sir. So the language that we can use for establishing a niche here, so we have, for example, a previous studies of strains of bacteria in soil have not examined. So that's how we write it in this way. And then we have such studies are unsatisfactory because, and then you explain, you support the statement. One question that needs to be asked, however, is, and then support your statement. Research on, for example, strain of bacteria in soil has mostly seen restricted to, and then you support the idea and so on. So that's how we establish the niche. Then move number three, which is the last move, occupying the niche. So this step is an explanation of how you are responding to the need for further investigation. So why do you want to continue studying or investigating your chosen topic? So you explain how your research addresses the need you identified in the previous step, and list your specific research objectives, questions, and methods. So we have here strategies for establishing or occupying the niche. Outline your purpose or your purposes of research, and then the questions and hypotheses, and then announce principal research findings, indicate the structure of your research process. Okay, some of this language to use for occupying the niche are the following. So state here, the purpose of this literature review is to identify or differentiate the different strains of bacteria found in the soil. Or you could say this study aims to identify or characterize the strain of bacteria found in the soil, and so on. So, and you can also say the evidence collected from the study demonstrates that, and then you support the statement, or this review outlines or examines, and then you continue supporting this statement. So I have here examples of Carl's model here. So we have in this first paragraph here, so this will be your first move, and then the next one is you have the second move here, and then the third move. So basically this will be your structure of your writing your introduction. Okay, next slide, sir. So now let's talk about writing your research title. So we will use the acronym FINER. So FINER stands for feasible, interesting, novel, ethical. So is your research title, research topic feasible? So when you say feasible, can it be done in a short period of time? Can it be done by a specific time frame or timetable? So that's feasible. So if your research title is feasible, then that's a good thing, and you can proceed to the next letter, which is the I, which means interesting. So let's say if your study is interesting, so how would we know your research title is interesting? So a research title is interesting if it drives a lot of questions. Like a lot of people will say, "Oh, your research is something that draws my attention, draws people's attention." So that's interesting. So more questions, can be drawn from your title, so that makes your research title interesting. And then the next letter is N, which stands for novel. So when you say novel, your research topic or research title is something new that has been studied, or maybe only few people have explored or pursued your research. So that's considered a novel topic. And then the next will be ethical, for E, ethical. So in doing research, there are ethical considerations, so which I will explain later further about the use of uh, animal models, kinds of plants, and then some protocols, because in doing research, um, there are a lot of forms that will be needed, especially when you are presenting your research paper in a conference or in a competition such as science investigatory competition. So it requires some ethical consideration, some like licenses or approval from ethics committee. And then relevant, the last one. So is your research title address some of the problems or the needs of the present time? So that's basically the finer. To evaluate, to assess your research title using that acronym finer. And then we have your sample like topics or areas you can explore specifically in life science. So here, number one, we have compare the effect of antibiotics on gram-positive and gram-negative bacteria. So you can grow your own cultures with agar and petri dish. And then, so for a sample procedure and more, so I put here the link of some of the project for bacteria science. So comparing the effect of bacteria or antibiotics on gram-positive and gram-negative. And then number two, run a bioassay to test for toxicity in water or soil. So this is also a good research topic that you can also explore. Number three, test the effect of ultraviolet radiation on bacteria growth. So this one is also a common research interest, especially if you want to investigate on how different UV light or UV radiation sources can affect the growth of different strain of bacteria. And the number four, experiment with plant genetics. So if you're interested in plant hybrids and crossbreeding, so this will be a good topic for you to start and explore. Number five, test factors like smoke or pollution that might affect transpiration rates for plants. So this one is also an interesting topic. Number six, investigate the effects of increased oxygen or carbon dioxide concentration on plant germination. So because we learned that oxygen is the product, the byproduct of photosynthesis, and carbon dioxide is one of the raw materials in photosynthesis. So we want to know if we increase or decrease the concentration of these gases can facilitate the germination of, let's say, seeds. So this one is also good topic, especially if you're studying crop science or crop production. And number seven, find out the differences in properties and effects of organic versus chemical fertilizers. So all of these topics, if you're going to evaluate that using the finer, there are really good topics to pursue, especially for students who are studying biology. Okay, next we have earth science, some topics here. So number one, explore methods of erosion prevention, test effects of different soil. So this is also nice topic to explore, if is especially here if you want to compare how does more clay compare to more sand. And number two, experiment with methods of flood management and containment. Especially in the Philippines, there are areas in the Philippines that are prone to flooding. So this is a good opportunity for for the students and teachers to explore different methods of management and containment of floods. Number three, investigate the effects of sunspots on weather patterns. This one is also interesting, but you need some sophisticated equipment for this. Number three. Number four, work with methods for forecasting weather. So back in 2000, if I'm not mistaken, 2018-19, we did something like this experiment and lab analysis. So we designed the IoT-based weather forecasting with some of my students back in the Philippines, and we even went to Korea and Singapore for this kind of topic. So this was a very interesting um, area to explore forecasting weather. Number five, test the concentration and effect of minimal minerals and pH in soil and water. So for example, you can use the water test strips and soil analyzer if you want to pursue this kind of topic or research area. Number six, determine chemical makeup of rain in your area. Test possible hazardous effects. Okay, next slide, sir. Next is physical science. So we have here experiment with the effects of storage, temperatures, and batteries. So batteries is also a nice area to explore. And then how this the lifespan or the life of a battery affects in terms of storage temperatures. Number two, develop improvements in battery chargers. Try methods of using solar cells, recharge batteries. So it's also a good area to explore, especially if the Philippines is in a tropical region, so we can harness energy from the sun. And number three, compare the bending strength and durability of different building materials. So I also was able to supervise students, especially in looking for compressive strength of materials back in the Philippines, especially the materials that were already considered trash, like plastics, seashells, and then they were added to the concrete hollow blocks. So they measure the compressive strength and the durability and also the ability um, to withstand different extreme temperature and pressure. So we did this number three with some of my students in the Philippines. And the number four, design industrial uses of magnets, test the effects of magnetic and electromagnetic fields on living organisms such as brine shrimp. So later we'll talk about brine shrimp as one of the animal models usually used in the lab. Number five, design a project in advanced robotic programming. So we also did this one in the Philippines, and then some of my students even went to LaSalle for a training. And back then, my students were first year, and then they had no knowledge about coding and programming. So they went to LaSalle for 2 months summer internship training there. So that was a very memorable experience for my first year students back then. And then number six, build a sensor moving advanced bristle robot. Yeah, so we also did some sensor robots experiments. Then for chemistry, we have so many we can explore in chemistry because we're dealing with substances, chemicals, and there are a lot that we can explore. So here, number one, experiment with types, effectiveness, and the impact on the nutritional value of preservatives in food. So you need some chemicals that you can use here, maybe some knowledge on nutrition, knowledge on um, nutritional facts can also help. Number two, test the mechanical properties and physiological impact of saturated, unsaturated, and trans fats. So if you want to explore in this specific topic here, so you will have knowledge about how fats are formed, and then what are the chemical properties and the physiological impact of these fats to humans, to our bodies. Number three, use the endophenol to test the effect of different cooking methods on the depletion of vitamin C in food. So there was a study that says when food is heated, especially food that is rich in vitamin C, the vitamin C disappears or evaporates. So this one is also a good way to test if different cooking methods can, deplete or reduce the vitamin C present in most foods. Number four, investigate the role of enzymes and yeast in the fermentation. So I remember my there were groups of students who went to UP Diliman and they studied enzymatics, especially the use of the enzymatics substances for diabetes. So they use different animal models for this study. Number five, experiment with different methods of water filtrations, purifications, such as solar distillation. And then number six, analyze the byproducts of gasoline, compare the efficiency of various octane levels. And then we have the environmental science topics here, or some of the interesting topics to pursue for investigation. So we have compare or develop methods of hydrogen production and storage for use in fuel cells, experiment with expanded use of solar energy, test methods for cleaning up and neutralizing the effects of oil in salt water with this oil spill cleanup kit. So you can use some kit here, especially lab grade kit. You can buy online or chemical companies. Number four, work with methods of processing, recycling non-biodegradable items, experiment with decomposition aids. So this is also a good topic to explore. And number five, experiment with design and function of wind turbines or water wheels. This one is very common topic for high school. And number six, test for harmful effects of pesticides. Test or develop natural organic alternatives. Test the effectiveness of common pesticides such as DDT. So these are different kinds of topics that you can do that follows the finer. Okay, so let's move on to the experimental design. So now you're ready. You have your own chosen topic of your interest, topics that you think very interesting and can help solve problems in our community, in our country. So now you're ready. You have your research design. So it is very important that you know some of the key terms in doing experiments. So in here, in every good experiment, there must be control group and an experimental group. So a control group and experimental group are two different parts of your experimental design. So in a control group, these are the thing. There are two types of control group. So you have the negative control and the positive control. So these are the things that you need to compare the results to the experimental group. So the experimental group is the group that receives the experimental treatment or test sample. And then the control group, the group without the experimental treatment, so it cannot affect the results. And then there are also other variables that you need to be familiarized. So a variable is any factor, trait, or condition that can be changed. So we have three common type of variables. You have the independent variable. So this is the variable that you want to change, or you want to manipulate, or you want to test. And then, so the result of this independent variable is your dependent variable. And then this is usually the one that is being measured. So the dependent variable is the result of what you've done in your independent variable. And then the control variables are the variables that you want to keep constant. So the purpose why we have control variable is to eliminate bias to your results. Next slide, sir. Okay, so I have here some questions because, like what I said earlier, in the scientific method, that it always begins with being observant uh, and then asking questions. So it's very important that when you write your research topic or research question, it must be measurable, and then we will use the different variables here. So IB stands for independent variable. An independent variable will be your DV. So for example, here, do tomatoes grow fastest under fluorescent, incandescent, or natural light? So you have that. This is a good question because we can measure uh, the parameter here. So for example, the growth. So do tomatoes grow fastest? So growth here can be measured, right? So we can measure the growth of the tomatoes. If we apply fluorescent, if we apply incandescent, or we apply natural light. So, but the question here is what would be our independent variable? So our independent variable is the variable that we want to manipulate. So the type of light the tomato plant is grown under. So that would be our independent variables, and the result, the dependent variable or the DV, the rate of growth of this tomato plant. And then the second question here, what is the effect of caffeine on blood sugar levels in intermittent fasting? So which one can we measure here? We measure the blood sugar levels. Okay, so that would be our the DV or dependent variable. So blood sugar levels are what we measure, and then what we manipulate are the presence or absence of intermittent fasting. Okay, and then next is medical marijuana effective for pain reduction in people with chronic pain? So what we actually measure in this scientific question, what do we measure? We measure the frequency of pain and intensity of pain. So what is the threshold of the pain? So that's what we measure. So that would be our dependent variables or the DV. And then what we manipulate is the presence or absence of medical marijuana use. Okay, so that's how we identify the variables in a research. And then we have another. So we're done with the peak. We're done with the finer. So that's how we evaluate the research title. Now we go to another acronym called PICO. The use of PICO. So PICO stands for the population, and then we have the intervention for the I, P comparator, and then we have the outcome. So population refers to the subject of your investigation. So in the case of tomato, growth of tomato, so our subject is the tomato. And then for the study of, let's say, earlier study about bacteria, so bacteria is our population. And then the intervention, what have you introduced to cause the change? So intervention is what have you introduced to cause the change, to cause the result. So remember, we have the IV and the DV. So the IV is our independent variable. What did you introduce to your IV or independent variable to cause a change, to cause the result, or the DV, the dependent variable. And in the comparator, this will be our the comparator will be our control. Okay, so comparator, so we have negative control, we have the positive control. So for the negative control, this will be so if we measure the negative control result, this will be the least value that we can get. And then the positive control will be the maximum value or measurement that we can get. And then the outcome will be in a form of result. So this will be the dependent variable. And then the outcome, usually the results of the experimental group. So, so we have here a good example. So this one a very common example of our PCOM. So for example, here we have the same plant. So let's say this is a, so let's just assume this is a picha plant, although it doesn't look like a picha here. So the independent variable is the variable that you want to manipulate here. So you have the liquid used to water its plant. And it says here that the one thing you change limit to only one in an experiment. And then you have the dependent variable. So the change that happens because of the independent variable. So this is the result because of what you've done to the independent variable. But there will be no results here in the dependent variable if there is no intervention. That's why between the independent and the dependent variables, there is the intervention that you introduced. So let's see what is being introduced here. So we have the control, everything you want to remain constant and unchanging. So for example here, type of plant used. So for example, for plant A, B, and C, we use the same kind of plant, we call that picha plant. So because the purpose of the control variable is to eliminate bias, and then the pot size also should be the same. So A, B, and C should be the same. And then amount of liquid. So let's say if you use only like 50 ml or 100 ml of the liquid should be the same. And then salt type, of course, also the same. So if you use loam soil for A, should be the same with B and C. Now, what are we studying or investigating in this specific illustration here? So the use of different, let's say, substances. So for example, here in A, use a substance difference in substance B and substance C. So therefore, we could say that we are testing three different substances, or we compare two different substances because the control variable is here in C. This one is distilled water. And then in A, it's a different substance and B is different substance. It says here juice. So probably, well, it's not indicated here what kind of substance for A, except for B, which is juice. So we can say that, let's say a soda A can help facilitate the growth of this peach plant. So A and B, and then we compare that to the result of the C. So C becomes our comparator. Okay, so going back to the relationship between independent and dependent variable, there must be intervention that introduced here, and then in the control variable we have the comparator, the negative and the positive result. Negative is the minimum value or measurement that we can get, and then the positive control is the maximum value that we can get. Next slide, sir. Okay, so now let's jump to the next slide. So this is very interesting because these kind of animal models are common used in the lab. So we'll start with the brine shrimp. So you see in the upper left corner, top left corner. So you see here the shrimp. Okay, so usually they are used for toxicity and also analyzing the if the substance is toxic or the substance is or a substance that you are going to evaluate has some contaminations, and then you measure the mortality rate of the brine shrimp. So they are basically shrimps, saltwater shrimps. And then you have the very, very common one is the lab mice or laboratory mice. They're used for extracting their blood or introducing chemicals or substances to lab mice. And it is said that 98% of the lab mice DNA is the same as the human. So lab mice is very common animal model to use in the lab, especially when we want to introduce substance or chemicals without the use of human. So we before we go to human or the clinical trials, we use the lab mice. And then on the left corner, the lower corner here, we have the zebrafish. So zebrafish is very interesting because there are so many experiments that we can perform. So for example, if you want to study on the effect of, let's say, very common in the Philippines, like whitening agent. So because there's pigment here, you can see pigment here in the zebrafish. So you can also study on toxicity and genotoxicity using the zebrafish. So there are a lot of literature. So I can, so I hope I can show you an example of literature or study that involves the use of zebrafish. So this one is very interesting. By the way, zebrafish is a native of in India and in Nile River. So it's they collected the eggs of the zebrafish, and then they allow it to hatch for like 5 days, and then they have the post fertilization. They only need one male to fertilize the eggs of the female. So like in an aquarium, you have 50 female and you only have one male, and that's enough to fertilize the the different eggs from the female. And then we have here the Drosophila melanogaster. So this is a common fly, fruit fly. So they are also used in different experiments, especially on analyzing the, let's say, you analyze the bacterial growth, you analyze the ability of these organisms to survive in a highly contaminated environment. And next would be the IC form. So IC stands for International Science Engineering Fair. So the Department of Education is under this organization. When you join Science Investigatory Project, you need to be compliant of these different forms. So here we have Form 1, checklist for adult sponsor, safety assessment form. Okay, so there must be an there must be a adult sponsor because especially the students are underage. So when they conduct experiments in the lab, they need to find um, a sponsor. Usually the sponsor are the scientists. They will be, and also the advisor will sign this form. And then there are a lot of requirements here. So if you click this form, so we have the link at the bottom here, and the details of this form can be found here at the bottom. And then we have Form 1A, Student Checklist Research Plan. So the research plan involves the specifics, the specific guideline and step-by-step process on how to carry out the experiment. And then also we have the Form 1B, the approval form. So for example, in the lab, you have uh, access to, let's say, different chemicals and reagents. So it has to be approved. And then a lot of scientists, or let's say lab technician, will supervise you, and then you need approval from these people. And then Form 1C is basically from which institution you conduct the experiment, because you cannot just perform experiment in the classroom when you're joining International Science Engineering Fair or Science Investigatory Fair. Classroom experiment will not suffice for you to join or be approved, because at least in the school there must be a ethics committee. And then for your research to be accurate, to be scientifically accurate, it has be approved and regulated by a research technician or research scientist, and the equipment that you use, the materials that you use has to be laboratory grade. So this one, Form C, usually the research scientist and lab technician will sign this Form 1C. Next slide, sir. Okay, so we have here another form, form to qualified scientist form. So like what I said earlier, a scientist has to sign your research before you conduct the experiment. You cannot, uh, because like what I said earlier, that a research must be ethical. So the qualified scientist will check if your experiment follows protocol or scientific protocol for you to be able to perform it in a research institution. And then Form 3 involves the risk assessment form. This one is also very strict. They will be looking into this form because they want to see if you are using biological hazardous chemicals, and then like tissue culture. So this form is very they're looking into it, especially during the presentation. And then a research paper can be disqualified if you're not following the what is required here in Form C, the risk assessment form. And then for Form 4, for example, but this one is not common because when you're doing experiments, most likely you're not using human participants. So it's rare to have human participants in an experiment. And then you have the sample informed consent statement. So this will be the form that your parents, the students' parents will sign, the school will sign, and a lot of people involved for the consent. And then here is more specific forms, specifically for your study. So for example, if you're using zebrafish, so zebrafish has a vertebrate, so you will need to use this kind of form. Or if you're using lab mice, so some experiments, some lab experiments will not qualify for presentation, especially if when they check your paper and then there's no signature or there's no approval, and then you use animal model. So there's what we call the um, the IUCOC, or the Institutionalized Animal Care Use Committee. So it has to be approved, especially if you're using animal model. Then Form 5B, vertebrate animal form. So this one is also very important, regulated in a research institution. And then for potentially hazardous biological agents form, that one also is very important. And then, yeah, so human and vertebrate animal form, tissue, for example, you collect blood samples, tissues, you also need to use Form 6B. Next slide, sir. And then if your study is a continuation, so for example, you studied last year and you want to continue your study or parameters, you want to explore more and continue more of your experiment. So this form is required, Form C, continuation project form. And then next will be abstract form with 21 categories. So it depends on your categories. So basically, these are the different IC forms that you will be using before you conduct the experiment. So it has to be completed, it has to be organized, because these are the forms that are needed during your oral presentation when you're joining different kinds of research presentations. Okay, so I guess you're now ready to write, right? So these are the tools. These will be your friendly tools, your guide, your your assistant in starting your writing journey. So I would suggest that when you write your research, you will use Google Docs because Google Docs is a web-based doc, so it will automatically save your file, and this will save you your time. So Google Docs is recommended instead of a Word document. And then we have the Quillbot. Quillbot is this is also your best friend when you're writing because in writing you need to. This is also a paraphrasing tool for you. Although some people don't recommend this, but I would highly recommend for beginning writers to use the Quillbot. So there is a premium account for this, and then there's also a free version of the Quillbot. Mendeley is to organize all your paper, your PDF file, your compilation collection of your research journals. It's stored here in the Mendeley. So there are a lot of tutorial on how to use the Mendeley. Turnitin is another software that can be very useful to you because whether we like it or not, some of our students, if you are teachers, some of our students they are getting the sources from the different online sources or journals. So some of their paper are copied from the internet. So Turnitin is a software that evaluate plagiarism. So it's a plagiarism checker, and then it will flag down some of the sources copied. Usually institutions, universities, they have Turnitin account, and 5 or 10% is usually the recommended like the percentage to allowable percentage for plagiarism. So if more than that, so you need to paraphrase, so you need to use the Quillbot. And then for Turnitin, once it flags down, there are recommendations that you can also see the websites, different sources where you get this information. So you can check again. So for example, if you're done checking your references or your sources, and then you go to Quillbot, and then you use the Turnitin again to check your plagiarism percentage or similarity index. And then finally, Grammarly is also helpful for you to structure your paper in terms of syntax, in terms of grammar, in terms of usage, because in scientific research, it's a different kind of writing. So it's a very academic. It's not the everyday English that we're using. So it's an academic English. So Grammarly can save a lot of time for you. Okay, so I think that would be my last slides. So if you have questions, clarifications, so I'm very much happy to answer all of your questions if there are questions.
Okay, so thank you so much, sir, for that very nice give us inputs. Okay, so it's like a walkthrough of a science investigatory project. Okay, so what's the specific target? Ano ba yung mga goals natin? Ano yung magiging dapat i-consider natin? Okay, so ang lawak talaga ng mga field of science na dapat natin pagbigyan din with regards to science investigatory project. Okay? So hopefully sa mga viewers natin, kung meron kayong mga katanungan or some clarifications, so at least nandito pa si Sir Saldi, sana po ay makapag maximize tayo ng message section natin. Okay? So habang ako nanonood sa iyo, Sir Saldi, parang sabi ko, malaking challenge talaga sa isang science teacher na mag-conduct ng science IP sa school.
True, sir. I would agree because um, I learned science in investigatory project for many years of engaging in different science investigatory like conferences, seminars, trainings. So I also was very blessed at the time back in the Philippines because I had a lot of mentors who are very generous. They were offering me like 10 years ago. I was
Mm,
invited
yes po.
to
Okay.
go on an internship training in University of the Philippines, and then I was recommended to attend a diploma
Okay.
course in Nagasaki
So yan,
University.
dear viewers, okay, so nakita natin paano na-present ni Sir Salde regards to Science Investigatory Project. So yun ay, di pa po yun ang totoong science IP, especially with regards to parts. Yun po parang guide pa lang. Okay, so ano ba yung magiging approach natin kung magkakandak tayo ng science IP sa school natin? What are the specific fields na pwede natin na pasukan? Some interesting topics na pwede natin pasukan din. Okay? So, how much more kaya kung papasok na tayo sa part by part ng Science IP. So, yun ay ano pa lang, introduction pa lang yun. Okay? So, again, very challenging talaga mag-conduct ng Science IP sa school natin. In my case, nakapag-conduct ako ng isang business lang. Kasi alam natin kung nasa sa field tayo sa DepEd, ang dami ding mga extension na trabaho ni bigay sa atin. Okay, so kaya sometimes ay din parang maubusan din tayo ng oras. Okay? Kasi dapat ito'y para sa akin kasi dapat kung mag-conduct tayo ng Science IP, first month pa lang na-introduce na natin para at least malaki pa yung oras yung may Google ng bata within the school year. Okay? So, ano kayang sir, mga magandang tips na ibigay mo para sa mga educators na kasama ko mag-conduct ng Science IP sa school? Okay, for especially may regular class at may science class din. So magkaiba yung mga classing student na doon na nandun sa science class at sa regular class.
Siguro what I can suggest or advise that para maging effective na maging research consultant, advisor is you must be a researcher yourself. Kasi doon ako din nag-start. So I first learn about
So,
the principles, the the in and out of research. I attended several trainings. I attended lab experiments, and then internship program. And then from there, marami na akong natutunan. So naging effective na ako as an advisor, as a coach. Kasi you cannot, 'di ba? You cannot teach what you do not know. So that's a
yes po.
basic tenet talaga for me.
Yes.
So kailangan i-equip mo muna ang sarili mo about research. You must conduct a research yourself. You will pair with teachers who are knowledgeable about research. You will form collaboration, and then from there, you can, efficiently and effectively and confidently share knowledge to your students.
Yes po, sir. Okay. So, sana sir ay hindi po ito ang ang huling ano mo po pagiging speaker po ng Science IP dito sa community ko po. Okay? So kasi ang dami ding science teacher na nanonood ngayon sa broadcast natin. Okay? So
Yeah.
hopefully po may guide mo din po kami in ano po paano intindihin yung part by part ng science IP.
Yeah, it would be my pleasure sharing my knowledge to the Filipino teachers and students.
Yes, sir. Especially sir, kasi I'm handling ngayon ng science class ng grade 10. So I'm planning to have one science IP, but gusto ko rin na equip din yung mga students ko from a certain session like this. At least they have this very masasabi nating fruitful talaga na input kasi galing talaga sa expert. Okay? So hopefully sir, we can still invite you if you're free po para
Yeah,
sa mga
anytime.
students ko po. Oo. Okay. So based sa sir, sa message section natin wala namang mga katanungan yung natin mga viewers natin. So if there are any, so say for instance mga late reactions na siguro baka nasa team replay and then nakapanood and then may kaunting katanungan. So consolidate ko na lang siguro sir tapos bigay ko lang po sa iyo ano yung mga concerns nila. Okay, so if you don't mind po.
No worries.
Yes sir. Okay, so since wala po kayong mga katanungan dear viewers, so alam naman natin it's Saturday, so siguro nagpapahinga pa yung iba, so baka mamaya pa manonood. Kaya di po masyadong active yung message section natin. Okay, so anyway, ang students ko ay nanonood naman and also may kasama sa masterclass. So siguro walang may bigay na tanong kasi still absorbing pa at this moment. Hindi pa nag-sink in siguro sa kasi even ako when I'm looking at wow, ang ang lawak pala at ang ano pala ng science IP. So ganyan pala yung pasikot-sikot ng science IP. Especially yung inputs coming from expert like Sir Salde. Okay? So hopefully sir sa next session natin yung yung parts ng IP paano ba atakihin yung bawat part ng science IP. So from rationale, so on and so forth. So sana po ay may guide mo din kami.
Yes, sir. My pleasure.
Okay, sir. So, and also, sir, if kasi ang daming mga educators na nanonood, baka may plano din silang invite kayo. Sana po ay makapagbigay din kayo ng oras. That if meron po, kasi there are cases na habang nanonood dito ay nagpa-plan din na baka pwede ka din nilang maimbita sa, say for example, mga LAC session sa science teacher, so sa mga malalaking school. So siguro mga more than 5 sections magkakaroon sila ng plano na magkaroon ng LAC session, so Learning Action Cell. So magiging topic nila ay Science IP at baka kayo po ang makuha. So sana po ay
Yeah, that would
may
be
guide
great.
mo din sila.
Yeah,
Yes po.
anytime, sir.
And also po sa school din namin, we're planning also po to have like that po na magkaroon kami ng LAC session sa science about Science IP. Sana with your presence po.
Yes, sir.
Okay. So, since wala po tayong mga katanungan dear viewers, so let's certificate po to Sir Salve. Okay, so okay. So, Digital Technician, okay, so Digital Technician, award this recognition to Sir Saldi Diaz Alima for sharing his invaluable expertise. Okay, during the 65th free webinar entitled An Introduction to Science Investigatory Project, held online platform via YouTube Live during the Digifil Technician 65th free webinar held on February 18, 2023 via YouTube Live. Okay. The session was conducted with certain hour of training Given this 18th day of February 2023, the DigiFil Technician, Cebu City, Philippines, signed by um, the three administrators, me, myself, Benavides P. Hironjo Jr., and my co-administrators, Norman B. Lanes and Arturo E. De Leon. Okay, once again, Sir Saldi, maraming salamat po. And any parting words po para sa mga students and para sa mga teachers.
Thank you
Okay,
very
go
much,
ahead
sir,
po.
for inviting me. This opportunity to share my expertise, my experiences in science and investigative projects. So I would say that I have a great fun sharing my knowledge, and I wish that a lot of teachers watching now and students will be encouraged more to pursue science and investigatory because it's a very exciting journey, I would say. You know, doing experiments, meeting a lot of scientists, and then also the fun that you can get, especially you will be, learning about science in real hands-on science in the lab, and then be able to present your results in a wider audience. That's a very fantastic experience. And of course, you have to publish your paper, and then you will be known around the world with your results.
Yes po, sir. Okay. So once again, Sir Saldi, maraming salamat sa napakalaking tulong na nabigay mo with regards to fruitful inputs para sa mga teachers and learners na nanonood ngayon. Okay? So God bless po and ingat po lagi and hopefully po marami ka pa pong matutulungan na nag-i-aim din na makukuha yung ano ba yung science IP in real life po. Okay? So, once again, thank you so much, Sir Salde. Okay.
Thank you very much, sir.
Bye-bye po.
Bye.
Okay. Sige, sir. Bye-bye po. Okay, so ayan, dear viewers, another fruitful session po na nangyari dito sa DigiFil Technician. So, that's very first time po nagkakaroon tayo ng specific discipline po sa different subjects ng tinuturo natin. So, now we're dealing with science. Okay, so Science Investigatory Project. So, sana po sa topic natin na Introduction to Science IP ay marami po tayong natututunan. Okay? So, again, this is a DigiFil. Okay. Palaging naghahanap ng mga content or topics na makatulong po sa function natin. Okay, bye-bye everyone and thank you so much.